So I tried to put together a bit of summary briefly to touch the hyperspec solutions that we have and focus then a little more on the SWIR hyperspec and actually discuss also the potential and options if combining both, because I think that has also quite a bit of power. Does this work? Yeah, so I think I don't need to tell you so much about the hyperspec imaging, but maybe just to remind that the PSI hyperspec cameras that we integrate into the phenotyping systems are the push broom hyperspecs, which allows us via actually the scanning over the object to generate the hypercube. And yeah, here maybe nice video of a potato plant when we scan actually throughout the individual wavelengths, and you can basically nicely see where are in the visible range the key zones where the plants absorb the light and where they start to reflect. And you know probably quite a bit, that depending on the zone or on the range of the spectra where we are, we can detect different interesting components in the visible range, the pigments in the near infrared, all what relates to leaf and cell structure. And then in the short wavelength infrared, the key interest are the water bands. But actually, it's also well known that there are more components, biochemical components, structural components that can be interesting, and more features actually that can be extracted even when combining actually the specific wave bands from the whole region, so combining the visible and the short wavelength infrared. And here I don't know if everyone is so familiar, or I should step a little bit in and just briefly touch the hypercube. What is actually the data result that we are obtaining with the hyperspectral imaging? Where we basically
we're over the hypercube. Yeah, we know how it works.
That's fine. So great. So the fact that we can actually work on the level of single pixels with the whole wave range gives us quite a bit of dynamics and flexibility in how we can work with the data and what data we can retrieve. But still talking, I think, a little bit about the hardware, which is essential. So you know, either we are using actually the visible range hyperspec or the short wavelength infrared, or combining actually the both. And that's the case of actually quite a few of installations already that have been done where we have the combination of both. So on a linear,
Sorry, so just quickly, so they both use the push broom, so the same technique. So if we wanted to, and we bought a shortwave infrared, we could connect it to our plant screen, like right next to our current VNIR.
Yeah, exactly. So both are using the same concept, same principle, same data format, same processing. It's just that if it comes to the spectral resolution, I mean, the SWIR is lower simply than VNIR out of principle, because there the optics is simply more complicated. But otherwise, basically, the principle is the same. Yes. And I think we have like 5 or 6 installations now where we actually combine both of them.
Okay, good to know.
Yeah. And basically, if discussing what it would mean, like to combine already existing VNIR with SWIR, it's basically just adding additional side linear arm that will be used. Actually, it's a pneumatic switch between one and the second one. So it's basically switching between the two cameras. So always the one goes to the middle position where the system is calibrated, focused, and scans over, and then the second one is swapped, and the same story.
Yeah.
So yeah, basically then you know pretty well the setup, because key is also the illumination. So basically the same type of illumination we use for the Venere hyperspec, so the halogen lamp.
Yep.
And now I think good and important to discuss a little bit the hardware specifications. So for the high specs, we're 1700. So the detection range is 900 to 1700. And with this, basically, we get a spatial resolution of like 1.5 nanometer per pixel, so 2 nanometer wavelength resolution. And basically, the systems are always calibrated to our standard imaging areas, which pretty much reflect the standard transportation trace that we use for the phenotyping. So the scanning area in the closed systems is 400 to 400 millimeters, but basically can be optimized. As you are scanning, you are basically then limited just in one dimension, in the X dimension, because we have actually both the cameras also on our field system, so the field tractor, where we can scan plots of 1-2 meters. So basically, in the Y space, we are basically unlimited. And that was for the SWIR. For the Venere, you know, and then each of the devices used for the specific detection of the structural features in the plants. And now a little bit touch the data acquisition, because as I mentioned, it's pretty much the same workflow, data acquisition workflow as for the Venere hyperspec, where key is basically tuning the speed with the integration time and the frame rate of the camera. So, this is always tuned for the specific device and the type of the camera. And for both the systems, always you have the radiometric calibration of the data, and this means always the dark white calibration. But this is standard irrespective of the type of the hyperspec, camera type. And for that, we are using either Teflon or Spectralon actually as the white calibration standards. And this is done automatically, basically on the level of each image acquisition. And I think this basically I just mentioned. Maybe one thing that we recently worked a little more on, and to be mentioned, that basically the result of the image acquisition, as you know, is the hypercube, which is the bill format with the HDR information. And with this, we are then going into basically the automated image processing, and the outcome is either the average spectra for each object, or calculated vegetation indices for each pixel object, the image data, and then all the statistics or data visualization options that you have. But what we added quite recently is that actually we can work also on the single pixel level, so we can export basically pixel by pixel spectral information, which can be used for some specific approaches actually, and some interesting analysis.
Yeah.
If you have a good computational power, I have to admit.
I was gonna say to do that would be crazy, but yeah. Depending on the amount of pixels.
Yeah. You need to have a good computational power, but we see that for some specific analysis, like example where we really had to go a little bit below the level of standard approach, where you take basically the average information, was when we worked with pine trees, where we had actually really few pixels that we could consider for the image segmentation, and basically for optimization of the segmentation and improvement of the thresholding, basically training the algorithms. It was pretty useful actually finally to use the pixel by pixel information to tune the algorithms. Okay, and yeah, basically here is just a brief description of how do we work on the data set. So you get the hypercube, then you need to specifically segment your images, and this is done based on some formula that you use for the segmentation. And here again, yeah, either standard formula, so you are generating some pseudo RGB images that you can use for the segmentation, or actually you can use a more advanced approach and search for with basically PCA, for example, for key wavelengths that can be then post hoc used actually for generation of specific indices for the segmentation. Yes.
Klara, just on the segmentation, because we were doing some fiber beams, and we're using your current just segmentation formula, which I think is like the MCARI one. And because the fiber beam has white flower, we're finding that's actually segmenting out as well as the background.
Mm-hmm.
Do you know if PSI is working on any kind of like, I guess, multi-segmentation where it segments maybe the outside of the plant and then can keep all the inner pixels or anything like that? Because we're finding it hard to analyze the flower because it keeps masking it out.
So, I mean, you can run the segmentation multiple times. You would basically just need to tune your segmentation algorithm specifically that you basically get rid of the pixels that are not wanted. And that's basically what I just mentioned. That yeah, we started to use, let's say, more advanced machine learning algorithms where we really like search for the key discriminant wavelengths for a specific trait or feature. Example, we did this in the field experiment, actually hyperspec data. We did a big experiment with calendula, and we specifically wanted to basically segment just the flowers out of anything else that is acquired in the image. And then basically the algorithm was really trained just for the calendula flowers.
Yeah.
And then we ran simply one round of segmentation only with the calendula flowers, and the second round segmentation only with the green part of the plant. So that would be the way, but you would need to basically go and, yeah, basically search for the key discriminant wavelengths that would be carrying actually the discriminant power specific for, yeah, the flowers and not for the rest of the object. And then you can run the analysis simply twice. And that's the way you would simply like threshold your image.
Yeah, I get you.
Yeah. And then there are also, yeah, options how, because depends on the resolution, but sometimes you can have actually a mixed pixels because you are on the border between like the green tissue and yeah, the flower tissue. And then we simply developed the tool that we can actually either include the mixed pixels or you simply completely exclude them from the analysis. So it's like more clustering and PCA searching for the discriminant wavelengths. I mean, if anybody interested in this, I mean, I could ask, yeah, either I can share a few slides later on if I fast search for it, or could just share you some results later on, because we are basically preparing the publication. Yeah. And open to discuss that. It would be great if the tool is used, because our vision is actually now to, because we did it now since like last a year and a half on a multiple types of experimental layouts. And we aim to implement these tools into the new generation or the new version of the Hyperspec analyzer. That's the plan.
All right, looking forward to that.
Just a quick one. I was just going to say that you guys have Rick Surridge at your disposal. He's the segmentation king.
Yeah, I know, but it does RGB images. That's the only difference. The VNR is a completely different kettle of fish.
I think you'll find that his capabilities will be right up your alleyway. Klara has also utilized him for some things, and she was also impressed. So
yeah,
definitely utilize him.
Rick is incredible, I have to admit.
We'll have to approach him.
Yeah. So let's continue, and I think I can just follow up further, and then I think, yeah, this probably I can skip because I just wanted to mention that the data structure format, as the cameras use the same principle, same data format, same calibration steps. It's pretty straightforward for actually for any type of for any operation for the data processing that you might need to know, because basically you just use the same pipeline. So same raw format, same data processed format, image format, etc. And yeah, the hyperspec analyzer I just briefly touched, and you know that you can use it for the advanced analysis, even if it has its weak sides. So that's why we are pushing now for actually a new version to be released.
Yeah.
And the hyperspec data export formats, as said, basically pretty much the same. The only difference is basically the list of indices that are automatically calculated, certainly. Because with the SWIR, basically, we have now predefined 2-3 indices for water content analysis, but there can be much more depending on what is required, what is needed, and also especially if VNIR and SWIR data are combined. Because if they can be combined, there can be much more indices and potentially interesting indices generated. So I think this I can skip because you know the general indices. I think this as well, some example from the visible range hyperspec. But I think I'll better mention there's something from our recent work where we try to play a little bit more around actually on depending on the task where we want to define the ripening stage or powdery mildew development. So basically with these tasks searching for specific wavelengths that would be discriminating between the different features we observe, and then actually generating basically indices that take advantage of these discriminant in wavelengths. For the short wavelength infrared, basically, if we speak about only about the short wavelength infrared, we are using primarily the water index. This means that the 1440 water band, and it can be used as a simple normalization band compared to the reflectance, the near reflectance band. Or there are actually multiple different combinations of the water indexes in this range that can be used. And here just a simple example of a pretty old experiment that we did, because you can calculate pixel by pixel the water index. You can use a color scale, so blue meaning high water content. I mean, looks quite fancy that the blue plants are full of water, and then when we are drying the Arabidopsis plants on the time scale, basically the water content decreases, so the plants are getting basically like pale. And again here for the segmentation of that's maybe one thing that is a little bit specific for the short wavelength infrared hyperspec, that one needs to keep in mind that if we have only short wavelength infrared and we have dry object on the dry soil, it's getting tricky to do any automatic image segmentation. So in this case, in past we used always combination with RGB or then combination veneer and SWIR. Because if the SWIR can't be used itself, SWIR data can't be used itself for the segmentation, for the effective segmentation, we can basically use, for example, combining it with RGB, and then we would use the RGB blend mask for the segmentation of the image, and the same mask would be then applied on the SWIR data so that the object can be effectively segmented.
So you can use your RGB, is that the RGB from the RGB camera or is that from the VNIR?
So if we have the combination veneer and SWIR, so near infrared with the short wavelength infrared, we use the RGB from NIR.
Yeah.
But if we have only short wavelength infrared, then we are adding additional small RGB cam that is basically always operated also in scanning mode. And we are using this RGB actually for the image segmentation.
Okay, yeah, so the RGB does the panning, so you get the same pixel size.
Exactly.
Yeah, because our RGB camera is very different to our VNIR. But yeah, if you do it like that.
Yeah, absolutely.
Yeah.
Yeah, so that are the two options, because we have few installations where we have solely short wavelength infrared, and then it's managed this way, because there are really cases when it's getting difficult for robust image segmentation only with the short wavelength infrared. Yeah. Okay. Yeah.
I also read a paper where they tried to determine like pathogen stress or heat stress using the SWIR.
Mm-hmm.
And they made a point that the calibration is really important. So they had different I think they used different temperatures of water or something to calibrate the SWIR. Is that something you've experienced with your setup, or?
That sounds a little bit special. They use different water temperature to calibrate the short wavelength infrared. I mean, the camera itself, the readout of the camera is not influenced by the outer temperature if we are in the declared range, which is 10 to 40 degrees.
Yeah, okay.
And if it's related to the response of the plant, there I have to say heat stress is, yeah, sure, will have impact, because you will lose the water from the plants under the heat faster than under control conditions, but otherwise I can't imagine how it could impact actually the actual data acquisition quality.
Mm-hmm.
Yeah, but yeah, maybe would be interesting if you would not mind to share the paper. That would be quite curious to have a look at.
Yep.
Yeah, so to move further, so I mentioned the potential in the near imaging, and with the short wavelength infrared, I have here one example from our own data. I will show some more data later on, where we did the determinant long drought stress experiment in barley over the period of 1 month, and at the late stage of the stress, we can pretty nicely see actually how the spectra in the blend, that's the average of the whole population, is discriminant between the control orange and the drought stress blends. Meaning that basically here at the 4,450, we have one of the key water bands actually that can be then actually captured and quantified either by the simple water index or different more complex water index parameters. And one nice example how we can use hyperspec imaging that I like to mention, that it's actually the optimized and customized setup that we did for Utrecht University. And that's specifically designed for actually fluorescent hyperspectral imaging, where we are using UV excitation light source to excite specific metabolites in the root of the plants. And now I absolutely lost the name of the metabolites.
That's fine.
No, I might. I will maybe remember later on. But basically the point is with the right excitation, we get a fluorescence emission. And with the hyperspec, we actually have such a good sensitivity that we can really detect also the fluorescence emission with that. So that was like to demonstrate that the application potential can go also beyond just standard reflectance indexes in plants. And now I have last two slides, and there I asked actually my great colleague Ivan to work a little bit on our potato data that we have from a large experiment we did last 2 years, where we did different types of stresses in potato, drought, heat drought, waterlogging. And we have quite a nice data set when we combined the measurement of Venere and SWIR. So basically for the course of the experiment, the 3 weeks, we basically daily captured both visible range and short wavelength infrared data. And what we can do actually is, if we have both the cameras, what we can do is to use the spectral span of the Venere SWIR data. So basically we combine the both cameras and the readout from both. So Ivan made this nice video where we basically read through the whole spectra that we capture from like 400 down to 1,700 almost. And when we have the whole span, basically it opens a little bit new options in the range of indices that we can calculate, and mainly the range of wavebands that we can then scan through in order to define the specific features actually in the plants. So what I have here is example just of 2 water indexes. So the standard simple ratio water index 960 versus 1440. And we can see definitely the parts of the object where we have actually higher water content versus the lower water content. Then we, basically we went through the literature and tried to test different water moisture indexes. Depends on the terminology, there is quite a bit. And basically scanning through, trying to define those that could be actually out of interest. And the same with actually indexes that, for example, go towards nitrogen and chlorophyll content. And there we actually see that when we start to combine actually the visible range and the short wavelength infrared range, we get actually more significant differences for some of the indexes than compared just to standard only Venere data versus SWIR data. And I think I have here, one more slide just showing then what you can do with the data, because basically now, maybe instead of the, how much is it, 800 wavebands we have in Venere, we end up, I think, with 1,200 bands that we have over the whole span. And that's the data set that we can then take into a next analysis, and we can use the transformation of the data search for the basically the discriminant components of the data set. And yeah, Ivan was so nice to take a little bit the data from our, so that's basically data from our potato experiment. And we took now just 3 time points where we have the beginning of stress, day 0, 2 days after the induction of the stress, like the drought stress we will be talking about now, and then we have just the last day, which is actually 1 day of recovery. And what we can do is then ask the simple questions. So tell me what are the wave bands, wavelengths that are the most significant ones, actually to discriminate or to actually describe the population of the data set. So then we have actually here, when we are at basically the day 0, when we have actually all the plants under the same conditions, we see that these are all the wavelengths in this near infrared, everything around 700 nanometers, which is pretty much basically the structural reflectance phase. And then when we look into 2 days after the initiation of the drought stress, you can see that actually we can already nicely cluster, the population nicely clusters to drought versus control. And if we ask again the same question, so what are the key wavelengths that discriminate the population, then we are getting to the wavebands of around 1960 nanometers. So meaning that we have actually then option to look at the data a little bit different, not basically going for the specific indices that can be analyzed, but we can like this define like the key wavelengths that can discriminate between the populations, for example, or the treatments, and take those and create actually new indices that would be key to characterize or discriminate our data set or our population of the data. And that's basically pretty much the approach we take now actually for the analysis of the hyperspectral data, because we see that basically working solely with the individual indices, we are actually losing quite a bit of information from in our data sets, clearly.
Mm-hmm.
And I think that's basically the end of my presentation.
Nice. Thank you.
It's really nice.
Pleasure.
Thank you, Klara. Do we have any other questions,
Thank
comments, any, I guess, discussion points, since we've got Klara here and got a few more minutes we could discuss things?
Sorry about
I was just thinking with you were mentioning basically the handheld option, right? So with that, I'm not quite clear of how that looks like, because is it similar to what Klara showed at the beginning of the presentation, or is it really a point? Because I think last time Eddie mentioned that they would be working with clips, or the pen style approach where you have to put it on a single spot of a plant. So I'm not.
So in the meantime, while Klara probably gets something up, did you, Ricardo, did you get my slides that I sent across and all the stuff to Anthony, or did I maybe forget to include you?
I'm sure you're in there. With all the quotes and everything, wasn't it? It had the quotes
I attached a comprehensive document on the handheld device. Did you get a chance to see that one?
No, so I haven't had
So I had a bit of a look at it.
Okay.
I was trying to work out exactly what the data that comes off it. Is it the same kind of build file, or is it, I guess it'll be more just like a standard data frame like you get from the polypens?
Yeah, then it's the polypen format, because then you are basically integrating the information over the spot
The one spot.
that is illuminated. So then you get actually the average spectra over the
Yeah.
area you measure.
Yeah, so you get the 2D data frame.
Yeah.
Okay. Yep.
And it's just that then you get it over the range where you have the detection. And this is either again, the visible range or the short wavelength infrared, or in the new spectrometer that we are now actually finishing and getting to the market, we get actually the span from visible range up to 2500.
Yeah.
Okay.
Yeah.
Yep. Thanks.
Just have a look at that document I sent across. So this is something Klara's been working on for a while. And again, the name is not final. That's just a working name because I need to call it something. Otherwise, I keep calling it the handheld device that does the full range. It's a mouthful. So yeah, have a look at that. And any questions that you have in regards to it, I'm sure Klara will have more details because she's done a fair few trials on different plants with it as well, and tested it and calibrated it, and tried different, optical probes with it as well. So we've done a lot of work on that device, and it's now basically ready to be ordered by various institutes. So, like I said last time, ANU is also very interested. CSIRO have been asking for it for the last 6 or 7 months, but we didn't want to send it over until we finalized and did the final calibrations. The policy is don't send a half-finished device to anyone, even if they say, "Oh, we'll be the first to trial it." It's never a good idea. So.
Sorry, sweetie.
Yeah. Klara, first of all, thank you so much for that presentation. I think it was really nice to just see what can be done from that PNRI as well as SWIR perspective. I just have a quick question. How easy is the data integration from, having said that we have VNIR in our rooms at the moment, and if we were to have the SWIR, using the same push broom mechanism, just wondering how easy would that data integration be, generated from both?
You mean, so basically, as I mentioned, the data set format is exactly the same as for VNIR. So we visible range. But if you speak about actually connecting the data sets, that's something that we are doing for the time being, basically in our Python or R script.
Yeah.
But this will be part of the new Hyperspec analyzer.
Okay, that's good.
So
I think
for the moment, we either work on each of the data sets.
Yeah.
And if we need to connect it, actually, not entirely, I have to say, because as part of the plant screen post-analysis option, we have a means already now to combine wavebands that are in the visible range and in the short wavelength infrared. This you can define already now. But if we speak about, like our advanced analysis, where we basically really want to screen throughout all the wavebands and search for the key features, for this, we have to use our own script that we have now, basically in Python written. This is not yet integrated. But if basically you want to, that you want to combine water band with NDVI band or whatever band in the visible range, this you can already do in the post-analysis. This is integrated, implemented, and can be basically straight away used for the analysis.
Cool. Yeah.
Thank
so
you.
not in the scheduler, you'll do it in the post-analysis and just
Exactly.
rerun it. Yeah, that makes sense.
And now my control question to Anthony. So, how often do you use post-analysis? Okay, you don't have to.
No, so at the moment, what we do is we literally load up the VNIR with all the vegetative indices. We've got about 20 on them in the scheduler. And that's what I've been doing my machine learning on. So literally just playing with traits and what do you call it? Classification modeling, but looking at all those vegetative traits.
Yeah.
I think in future experiments, I do want to go back because I want to recompute at a different trait mask. So I can just kind of draw a new bounding box and do more localized analysis at specific areas of the plant. That's where I'll go back into that post side of it. Yeah.
Yeah.
And I mean, in general, we are really what you introduced last with the binning and the PCAs to identify the wavebands that are most telling for your treatment and your plant. I think we are definitely wanting to move to that type of analysis because, I mean, we are setting up the database in the background so that we can combine the actual classification models that our cultivators provide or metabolite data that we get from our metabolomics platform or anything like that with the spectral imaging. So I mean, that is sort of our long-term, I mean, we are not quite there, but it's nice to see that you have already looked into this and that it really, that whole approach works. Sort of that's really nice and reassuring to hear because we were thinking along those same lines that we would go into that way because as you were saying, the vegetative indices are not really making full use of the data that the instrument provides. So yeah, that's nice to hear that you are also doing that.
Yeah, and I think exactly, I fully agree. And I think you mentioned biotic stress, and that's something that is definitely absolutely interesting. And there you certainly need to go this way. And I think there it's interesting then to go a next level where you are basically really thresholding for like specific parts of the tissue with the specific hyperspectral profile.
Yeah.
And when you mentioned the metabolites, maybe I would still share one more slide.
You found it?
This one. Which is, does it work? Yeah. Which is actually the thing that we do now. We are actually done. We are going, we have the manuscript in preparation. That's basically when we did our field experiments with calendula that I touched a little bit. And there actually we really combine actually multiple sources. We combine the hyperspec data sets. We had a manual polypen type of measurements done as well. So that was our like validation proof. And then, some RGB images combining actually different inputs with metabolomics. That was measured on the population. We had like 40 different cultivars of calendula. And we are actually, in respect of the classification, I mean, the model works basically perfect, almost too good, I would say, if we combine actually the inputs. For the metabolomics, it's certainly a little bit more tricky because there you need to have really good data set so that there is enough of metabolomics data measured. So there we are a little bit on the limit because we did not have it measured for all the 40 lines, just 8. But the initial results are quite interesting and definitely promising.
Yeah,
so we will be
exactly what we're doing as well.
Yeah.
So it looks great.
Yeah.
I think people are relying too much on an indice as opposed to actually joining everything that a phenotyping camera can produce and use that for this predictive modeling.
Because you can simply improve the prediction accuracy with that. Not always. Sometimes for some cases we see that actually the hyperspec data is more than enough we need. But for some specific lines, like we had very tiny flowers, and there, as we are simply remote, we have the sensors on the tractors, we have really few pixels, and then it's getting tricky. And then in this moment, when we use actually the other features, it improves the model. And with the metabolomics and hyperspec, actually, together with people from Ornella in US, with the Oak Ridge lab. There is a publication on poplar, and metabolomics actually included as well. So that's coming out pretty soon. So definitely, I think there are now more publications coming finally. So that's great.
And Klara, has anyone used the PSI hyperspectral in cannabis at all? Do you know?
In cannabis, no.
Yeah.
No, you are the only one. This I can tell you. So whatever comes out, I think would be really appreciated. So
as an experiment,
I don't know
right
about
now,
anyone.
so
Yeah, because I think your example, your example with the root imaging and the fluorescence. I mean, how does that work? So you excite with a UV laser, and then you measure what comes back with the fluorescence camera. Is that how you do it?
No, it's measured actually with the hyperspec in this specific excitation or emission range.
Okay. Yeah.
Because with the fluorescence camera in this range, it's tricky and the signal is not enough actually to be captured.
Oh, okay.
Yeah. So yeah, that's one application, and I will just now have a look at the name of the metabolites.
Very curious, because you mentioned it is associated with SWIR, and so it has to be connected to the moisture content as well, right? So yeah, so very curious to know what that metabolite is.
No, it's not related with the moisture, actually. Sorry,
No?
so this was maybe a bit confusion. It's not the moisture. If you give me, I will get to that point while we can still discuss. Yeah.
For the moment, you only have the UV for excitation, so you don't have any other.
Yeah. So basically, a specific illumination source, just UV, homogeneous UV illumination. And in this,
that would be interesting for cannabis, because you have the trichomes quite exposed, so it's really a different focal plane to the rest of the plant, and then they do absorb in the UV, so that, I mean, that would be
hit it with the UV and then take the image straight afterwards.
Yes, I'm not sure. Yeah, but yeah, might be.
Yeah, this I fully agree. Basically, even here in this setup, it was quite a bit of optimization and tuning to make the system basically robust enough, because one simply needs to remember always that you want to do automated image analysis. Yeah. And there always contrast big enough is the key issue. But I could imagine that if whatever with your hyperspec analysis, you identify some specific wave bands or ranges which are key signatures for your specific metabolite, then I think it's relatively straightforward to develop a handheld device that would be simply customized for this specific application. Because then you can easily implement different illumination sources, detection in a specific range, etc.
Yeah.
Would your handhelds be the one that you're working on? Like, could that be used?
Yeah, once you tune it, I could imagine that could be the case. Yes. Yeah.
Yeah. So currently the handhelds will produce the data, then you got to look into the data to find out what's the specific wavelengths you look for, but then you need to create a new device that looks for that exact wavelengths, pretty much turning a hyperspec into a multispec.
Yeah, or multispec as well.
Yeah.
Or you figure out that basically you need just two ranges, two specific ranges.
Yeah.
But that's always tuning. So I know already coumarins are the metabolites. Sorry.
Okay, yeah, that makes sense. Okay.
Yeah.
Yeah.
Yeah, exactly. So yeah, as said with the cannabis, but the things can be also tuned and tested.
Yeah.
As said with the coumarins, we also had to tune and optimize, define exactly the ranges of the detection where we need to go, tune the excitation light source. Homogeneity of illumination is key always.
Yeah.
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 Yeah, because
Great.
like Ricardo was saying, with the cannabis thing, a lot of it revolves around the UV area, but we're curious to see what the actual VNR can come up with and maybe combining with the other cameras, similar to what you did. Then I want to do like localized modeling around the actual flower as well, rather than take the plant average and to see what the results come back with. But we will be doing metabolite profiles, so we will be doing some prediction modeling. So I'm very excited about it.
Yes, sounds super cool actually.
And we'll let you know too.
I would be really curious to know, would be wonderful. So maybe you should really plan a visit again, and then we could discuss a little bit more on the data.
Yeah.
That would be really nice.
Be sweetie.
And I mean, if you want to discuss the approach, we, for example,
No,
and
definitely.
yeah, if you would want to discuss the approach we use now for the metabolite predictions, I mean, open to yeah, have a short meeting. We can share our results and discuss.
Yeah.
Yeah.
So for that one, you actually have a UV camera that you hit the plants with and then take the image straight afterwards. Is that correct?
That's your question, whether we have the UV camera for that.
Yeah, for that metabolite one you did, you actually have a UV light source and you spray that onto the plant and then you take the image with the VNIR straight afterwards and try and catch the other side of the excitation.
No, actually, you have the light, you have the camera, light illumination, camera focused, and scanning. That's exactly how you do it. Yeah.
Do you need a filter for that to cut out the UV, or is it?
No, for the UV, that's not a problem. The problem was that initially we tried to use actually like a broadband, so to have one light source for everything, but that does not work for this specific application. So in this given example, actually, we have two different light sources that are being used. Either it's the UV with the hyperspec, or it's the white light that you use for the rest of the spectra.
This might be a stupid question, but our current halogen light, can that be swapped out for the UV glow?
Theoretically, yes, but you need really homogeneous illumination.
Okay.
So, you know, we spent quite a bit of time actually tuning the light source for the hyperspec, because as you know, the halogen lamp, actually the issue is that basically you don't go below 420. And the hyperspec has detection from 380, but we just don't have the light there. So that's why we basically don't get into any metabolites that are being excited in UV. So this is still basically ongoing attempt testing different lights. We try to combine with LEDs, but the problem is that you really need to focus light.
Yeah.
You need quite high intensity and very homogeneous. In other means that every pixel gets the same, ideally.
Yeah.
And that's not, we are not yet there. Tested all different approaches. So if only UV, that's easier. If you want the whole thing, then it's getting more tricky.
Yeah, and even other companies let you do two separate cameras. You have a UV camera and then you have your VNIR or your SWIR. You'll never see it completely combined.
Yeah. But yeah, let's see. Technology evolves so fast. So let's hope there will be some new tools coming.
Awesome.